dangerous disease, dangerous virus. So nowadays we are getting vaccinations also against it. So this entire hepatitis family, hepatitis, hepa means always refers to the inflammation of the liver. So anything which is referred to or anything which related to liver, we always call it as hepa. Okay, like how we say cardiac, cardiac is referring to heart. So similarly, hepa refers to the liver. So hepatitis describes it basically describes the inflammation of the liver. Liver gets infected, liver gets inflamed, liver gets swollen. Now it can happen because of extreme consumption of alcohols or drugs. Sometimes it can happen because of autoimmune diseases where your own cells are being targeted as non-self, right? And sometimes metabolic diseases where some of the molecules which are not getting metabolized properly in the liver, they might get accumulated and cause problems, and viruses. Now, when we say about viral hepatic inflammations, that is viral hepatitis is basically the hepatitis virus is the one which affects the liver. So liver inflammation happens, and hepatitis can be are classified into different names. You have hepatitis B, A, B, C, D, E, F, and G. Whereas of course F is now F and G are not much significant. F is found to be a mutant of B viruses, and they have different types because if you see their genomic structure, not all of them contains DNA, and not all of them contains RNA also. So as I said. They are different in their genomic structure and as well as the infectious diseases, but of course all of them are in basically causing the inflammation of the liver, but they are classified into different names, hepatitis B, A, B, C, D, etc., because as I said they are all not of the same genomic material. So if you see hepatitis A, C, D, E, all of them are RNA viruses, whereas the only DNA virus which has been found is hepatitis B virus. Okay, so that is why that separate family also is being considered for that. Now, the viral hepatitis can be classified into two types of inflammations or two types of diseases: acute hepatitis and chronic. So when you say acute, acute always refers to any disease, and it's an acute disease. When you say acute disease, that means the disease is sudden. The disease is sudden onset of the disease, but the disease will not last for more than 6 months. So it's very short. Okay, but whatever during short duration, it can cause whatever injury or whatever problems it can cause. Chronic hepatitis, chronic means lifelong. So mostly when you say chronic, it actually refers to lifelong. We don't know exactly. It will definitely be more than 6 months. You can say it may be 2 years or 5 years. It can be lifelong. So hepatic inflammation, which is more than 6 months. Okay, and prevalence, if you see, as I said, more than 2,000 people have been infected with HPV, and still we are having nowadays HPV vaccination is also becoming more and more compulsory. Now, coming to, we are talking about hepatitis B. We are not talking about any other viruses. We'll talk about hepatitis B virus, which is enveloped virus. You know what is enveloped virus? Which contains the lipid combining lipid composed layer, and it contains double-stranded DNA. Okay, so and this is put under, as I said, this is the only DNA virus which is the virus among hepatitis, and it is put under a family called as hepadna virus. Now, how do you remember this hepadna virus? Here, if you remember, hepa is for hepatitis, DNA is for DNA. So hepatitis DNA virus, that's the family name. Okay, and it infects your liver and it goes and sits in replicates in the liver cells, which are called as hepatocytes, and the immune reaction is activated. So this is more or less like a coronavirus, where the corona, it goes and enters into your lungs and starts inducing your immune response after a delayed response. And what happens, we get a cytokine storm. That means your body starts producing excessive of inflammatory materials, and that results in the damage to your organs and tissues. Okay, so coming to structure, it's a DNA virus. It has a very compact genomic structure, and it's mostly the double-stranded DNA virus, a circular, partially double-stranded virus, and replication here occurs by reverse transcriptase. Okay, and it is a small circular 3,200 base pair size, and it codes for 4 types of viral proteins. So it is small, circular. It codes for 4 types of viral proteins. I'll show you the structure, and I'll also tell you about this particular HBS antigen. Yeah, so this is a basic structure of hepatitis virus. So you can see this is an enveloped virus. It has an envelope. Obviously, the envelope has proteins. Now, that protein is called as hepatitis B surface antigen. SS is smaller here. And this is what you normally get nowadays when, if anytime, if you have got a chance to, yourself getting vaccinated, or if you've seen somebody of your relatives getting vaccinated against hepatitis B, you could see there the vaccine bottle or the vaccine thing. They would have been recombinant hepatitis B virus, HBS antigen. What is it? We are injecting the patient with the surface antigen. So we need only to stimulate the immune response. That is why we are only inoculating the patient, or we are vaccinating the patient with hepatitis B surface antigen. And then followed by, it has a capsid coat, or the protein coat, and then followed by your the DNA, double-stranded DNA, circular DNA, partially circular double-stranded DNA. You can see here one area of that DNA is singular. That is why we call it as partially double-stranded DNA virus. So it's not complete because one area of the DNA is single-stranded. Okay, so it's 42 nanometer in diameter, and it's composed of 27 nanometer nucleocapsid, and which contains HBC antigen. HBC refers to core antigen and contains an envelope which contains HBS antigen. So for vaccination preparation, we are utilizing this HBS antigen. Okay, so it is having a double-stranded DNA. Okay, so there are three antigens which you can see here. One, we have already, I have told you already, hepatitis B surface antigen, so HBS antigen. And the second one is HBC antigen, that is your core antigen, the nucleocore or the nucleocapsid. And inside that, you will find few more proteins which are present, which is called as hepatitis B E antigen. E means referring to the secreted, okay, secreted protein. So HBS S refers to surface, or that is basically your envelope protein, envelope antigen. HBC antigen, that is your inner core protein, that is basically your nucleocapsid protein, and HBE antigens again further inside, which is called as the secreted protein. Okay, so this is a cross view of how the hepatitis B virus will look like. Now, coming to the replication. So this is an animal virus. Again, this has a replication which is different from a bacteriophage or a plant virus. So here also the virus enters into the cell. The HB virion binds to the receptors. Where does it goes and binds? The hepatocytes, because that is a site of infection, the lung cells. Okay, once it gets attached to the lung cells through its receptor molecules, through your HBS antigens, it enters into the cells and reaches the nucleus. What happens? The virus attaches to the liver cells, the nucleocapsid. The nucleocapsid means the nucleic acid plus the capsid enters in the cell, reaches the nucleus. There, uncoating happens. Okay, after uncoating, that means your capsid gets removed, and once the uncoating happens, reverse transcription occurs. Okay, so reverse transcription in the sense where the DNA is DNA is transcribed into your mRNA, and the mRNA is further reverse transcribed into DNA. Why? Because the DNA is the one which has to get packed up, right? So I'll show you. Yeah. So first the virus attaches to the liver cells. You can see the virus attaching. Then it gets engulfed into the liver cell. I told you most of the animal viruses, complete virus gets engulfed by the endocytosis process. After the endocytosis process, it gets degraded, and the nucleocapsid gets released, and the nucleocapsid, the capsid protein gets released, and the DNA enters into your nucleus. Okay, so the DNA of the virus particle enters the nucleus, and depending upon what is the work, that is the nucleus, when it enters the nucleus, the DNA of the virus start producing the mRNA. So you can see here, this is the nucleus. The DNA is producing the mRNA. Okay, this is the mRNA, and the mRNA will undergo translation to produce all sorts of proteins, the surface protein. Okay, so it's surface protein, and at the same time, another set of mRNA is that is from the DNA itself. You will have the mRNA which is being produced. So then, one set of mRNA will be utilized to produce surface proteins. One set of mRNA will be producing the core protein, and one set of mRNA will get reverse transcribed into your DNA. So this is what happens where reverse transcription happens, and the DNA gets produced, and the DNA gets assembled. The DNA gets copied. Okay, the DNA gets copied into this thing. So here, that is why the reverse transcription is asking for happening, because DNA gets converted to RNA. RNA does all the translation, everything work, and then RNA also has to get transcribed back, or it has to pass the information to produce a new copy of DNA. So that is why reverse transcription happens. Once everything is produced, packaging happens, and without lysing the cells, the hepatitis B virus comes out. So you can see the assembly of the virus. The virus is getting assembled by utilizing the capsid protein. The DNA assembly is taking place, and once after assembly, it can get exocytosed from the host cell. So you can see the virus is getting exocytosed from the host cell. We have already discussed about this exocytosis and endocytosis. Exocytosed from the cell without causing the lysis. So once it goes outside, it will go and infect the other cell. So this is normally how we observe under the microscope. It kind of gives a ground glass appearance because these proteins tend to stick together. Okay, so that is what we observe under electron microscope. So that is the simple way of explaining how. See, this is how you can how hepatitis B virus multiplies. So you can see the virus is there. Virus attaches the host cell. Fusion takes place. Dissolving of this particular area takes place. The viral nucleocapsid enters inside. The viral nucleocapsid, the capsid gets degraded. The DNA is taken. Again, DNA has a single-stranded region, and then it enters the host cell. The DNA gets transcribed into RNA. RNA gets translated into the proteins. Okay, so transcription, then translation into proteins, and once after translation, the mRNA also will get transcribed back to the DNA by the reverse transcriptase procedure. And once one strand gets synthesized, another strand also gets synthesized. So we'll have a double strand DNA, and then when it comes out, it will just come out through the exocytosis process, taking up all the envelope proteins, whatever envelope proteins is there, it will take it out and come back. Okay, so this is the life cycle of hepatitis B virus. Similar to your tobacco mosaic virus, only thing you need to remember especially here you have to remember the surface antigen proteins. So S antigen is very much important. Mode of transmission normally happens by different methods. It can happen through sexual contacts between people who are infected, parenteral, that is through blood, syringes, instruments, etc., and perinatal through mothers. So if the mothers are HBs positive or HPV and positive, they're able to transmit the infections to their children. And we'll not go into stages of disease. So with respect to your syllabus, you don't have, because this is not a medical paper, we're learning only about the cycle of the hepatitis B virus. So we'll stop here with respect to the life cycle of the hepatitis B virus. So any doubt in this part?